So, adaptation in the respiratory system, primarily due to the adaptation when we go to altitude, the adaptation when we suffer from hypoxia in its various forms. One of the sort of overarching concepts to keep in mind as apart from all the things we've learned about the control of the respiratory system, where we now have all the knowledge we need to tackle what happens when we go to altitude, what happens when we have prolonged hypoxia. And remember that the hypoxia system response is a fail-safe system. You can think of that in terms of the carotid body, which is our main responder. Remember Torrance's idea. This is a CO2 receptor that's highly sensitive, but it is calmed down by oxygen. So when we lose that oxygen, then we really potentiate the carotid body response to CO2. When we look at the control of cerebral blood flow, because after all, the main thing that we're trying to do when we cope by altering ventilation and various other factors is we are trying to keep the brain alive and well. So we have to increase cerebral blood flow. How do we do that? Again, I published a fail-safe model of how that works, so that when hypoxia occurs, the smooth muscle regulation of cerebral blood flow begins to fail, and that increases the cerebral blood flow. So, what are we going to look at? We're going to look at the ventilatory response to hypoxia. I referred to that HBR. People glibly state that, and I don't think they really understand what they mean. We'll find out. Hypoxic ventilatory decline, what is it and when does it occur and why does it occur? What happens if you apply an intermittent hypoxia, the kind of hypoxia experienced by OSA patients? And we did an experiment that I showed you last week about that. Then if we prolong hypoxia, such as going to altitude or suffering from a pulmonary problem, what happens then? And finally, I would separate out chronic hypoxia, where you live for many years, or you've been born and live at altitude in hypoxia. So to begin with, the hypoxic ventilatory response, HVR, what is it? Well, here's our ventilation versus PCO2 graph that we've already become familiar with, the metabolic hyperbola here. And we've done 2 rebreathing tests. Here's our wakefulness drive drawn in, and here's the hyperoxic. In other words, this is the central chemoreceptor response. And this is mostly where we go. If we go from a PO2 of 100 to 150, it doesn't change very much. So here is mostly where we lie. If we get hypoxic, then we bring in the contribution of the peripheral chemoreceptors in the carotid body. And so if we maintained an isocapnia here at this level, which is +2 above the normal resting equilibrium point, then if we went and gave a hypoxic stimulus, what would we see in the terms of an increase in ventilation? It would go up this much according to the red arrow. But what happens if we decided, we'll keep it isocapnic here at 40? Well, the arrow is going to be smaller. What happens if we were a bit nervous and in fact we were down here riding on our wakefulness drive sort of thing because it rose up here or some other aspect? Again, the difference here is going to get smaller and smaller. If we were hyperventilating down so that we were off the metabolic hyperbola, then nothing would happen if we got hypoxic. So let's look at that in a slightly different way. This illustrates, as we were talking about, suppose we were asleep and therefore we don't have a wakefulness drive, we have the ventilatory recruitment threshold, which becomes the apnea threshold. But here's the difference between our hyperoxic rebreathing line and our hypoxic rebreathing line at say 50. That would be our hypoxic response to 50 millimeters of mercury arterial PO2, but it depends on the isocapnic level. And so if we're isocapnic at 43 and we say, we'll do this the standard old-fashioned way of just by decreasing PO2, and we'll record what happens to the ventilation, but we will keep it isocapnic at 43. And we'll see this rectangular hyperbolic response. We can straighten that out in a way by plotting versus the saturation of oxygen in percent, and that's this dotted green line here. But suppose we had decided we would keep our subject isocapnic at 47, then we'd get a much different response. We'd get much more of a response, whether we measured it as the rectangular hyperbolic response or as the linear response to SO2, or as the difference between two steady states as we have shown here. So it depends very much. Lesson 1, the hypoxic ventilatory response depends on the isocapnic CO2 tension. And as far as I'm concerned, only rebreathing tests actually measure the chemoreflexes. The argument went on for quite a few years, and finally we had Frank Powell with his little editorial review of the paper that I published, and we finally have convinced him and others, I think, that you must be very careful when you're talking about the hypoxic ventilatory response and how you measure it. There is only one way as far as I'm concerned. That's using rebreathing tests. Now, what about hypoxic ventilatory decline? What is that? Well, suppose we keep the subject isocapnic at their resting level of PCO2, and then we make them hypoxic down to a level of 50 millimeters of mercury PO2, and we do this for 5, 10, 15, 20 minutes. Ooh, what happens? Well, let's take a look. The ventilation response goes up at first. These are breath by breath points. We put a line through them. And then it declines. And yes, we have published this. And so that is hypoxic ventilatory decline. It's been well known for many years. And indeed, if we take a look at Peter Robbins in the Oxford lab, we find that he's got several subjects here, each of them showing the hypoxic ventilatory decline, because they've kept PCO2 constant and they've given 20 minutes of low PO2. And so that is what is happening. We get an initial brisk response to the hypoxia that we've stimulated ventilation with, but then it decreases and falls away. Why is that? Well, the lesson is that the hypoxic ventilatory response itself is labile, let alone having to measure it at an isocapnic level. And so one of the ways that we looked at it was the effects of intermittent exposure to hypoxia. This is an example, as you'll see, of the potentiation of the carotid body. How did we do that? Modified rebreathing tests, as we've described. On alternate days, they were hyperoxic for central only and hypoxic for the sum of central and peripheral. We didn't want to do 2 tests in a day, we just did 1, before they underwent a 20-minute exposure to isocapnic hypoxia. And then we measured it again afterwards and took another rebreathing test then. And we repeated this on several subjects for 14 days. It's published here. And what did we observe? Well, rather interesting results. Here's ventilation versus PCO2 for this particular subject. And this is the hypoxic rebreathing test. And this is before hypoxia. We had a ventilatory recruitment threshold just over 35 and a nice response here. And then after hypoxia, we tested again. The ventilatory recruitment threshold has increased. The response itself seems to be very similar in terms of its slope, in other words, its sensitivity. So we can't say that it has sensitized, but what it has done is potentiate the response, because for any given isocapnic level, we now see we not see this much, we see this much. In other words, we've had that hypoxic ventilatory decline. When we started, we had this much response at this isocapnia. When we ended, we had this much. What happened over 14 days? Again, this is an isoxic rebreathing test at a PO2 of 50. So we haven't seen anything happening for the central key receptors with our hypoxic tests, rebreathing tests. Ventilation versus PCO2. On day 1, we had the ventilatory recruitment threshold here and this kind of nice linear response as we rebreathed. Then, after 14 days, the same test, rebreathing in hypoxia, the ventilatory recruitment threshold is decreased. And in this particular subject, AM, the slope was increased, not all subjects. And in fact, that wasn't a group response that was significant. But this VRT decrease was a group response that was significant. Let's take a look at the group of 7 here over the 14 days and see what was the ventilatory recruitment threshold doing over those days of exposures. Here's where we start before we expose to the 20 minutes of hypoxia, and then after a little bit of hypoxic ventilatory decline because the VRT increased. But that gap widened so that by the end of the 14 days, we had now dropped our ventilatory recruitment threshold over those 14 days. And then after our 20 minutes of hypoxia, the ventilatory decline was greater, and so it more or less went up to what it usually does. But this is the part that's interesting. Over 14 days, we had a reduction in the ventilatory recruitment threshold. That means that we've got an increased carotid body tonic activity. We're not more sensitive to CO2, but we've shifted it down, so there's an overall increase in carotid body activity. So here's another case where we have intermittent hypoxia, and we talked about that in obstructive sleep apnea. And I refer you to this figure from one of Dempsey's reviews back in 2010. This is a very nice figure to illustrate obstructive sleep apnea. We have the changes in oxygen saturation that occur so that we have our intermittent hypoxia. How did we get that? Well, that was by obstructions, because you'll notice here, this is the esophageal pressure, and this is the flow. And something has obstructed, and the effort to breathe has caused a decrease in the esophageal pressure as we try and suck air in more and more and more. And finally, we do, and we get some flow back, and we return. And this, of course, pattern repeats. This is obstructive sleep apnea. But here we are. What does this do, apart from potentiating the carotid body, tonic activity, which I just showed you? The evidence here, as I quote from his paper, is strongest to support the daytime systemic hypertension as a consequence of severe OSA and less conclusive efforts on other things. But here we are, daytime systemic hypertension. This is a problem. And we have problems with the heart as well coming through, cardiac arrhythmias and so forth. So what could we possibly do about that? It's the carotid body tonic activity that has increased. We know that that's going to increase the sympathetic nerve activity. And so here's a treatment for hypertension that has come into prominence recently. An editorial by Julian Payton, my good friend, targeting autonomic imbalance in pathophysiology. Is the carotid body the new nirvana? So it has the powerful reflex responses. The remedial actions when it is activity is removed or diminished. The carotid body therefore appears to be a target to remove, at least in the rodent models, as is in 2017. Now we switch to, again, he's involved with Nowinski's paper, 2021, bilateral carotid body resection, a novel treatment for sympathetically mediated diseases. The carotid body is more active. Well, let's take it out. But should be used with caution due to the risk of significant desaturation, even during mild hypoxia, equivalent to that experienced during long-haul air travel and high altitude. You've lost your ability to respond to hypoxia. So, extremely dangerous. Hopefully you've lost your high blood pressure too, but is there a better way? One of the ways that seems to occur to me that nobody has investigated as yet is, I've just shown you that repeated 20 minutes episodes of hypoxia can increase and potentiate the carotid body. How about repeated 20-minute doses a day, once a day, for hyperoxia? Doesn't have to be very high in order to shut off the carotid body. Don't go too high because then we generate various hyperoxic problems. So just enough to shut down the carotid body. Would intermittent hyperoxia do something like shutting down the carotid body in people with hypertension? Anyway, a thought. What about prolonged hypoxia? This again is an example of the potentiation of the carotid body. How did we investigate that? We did pairs of rebreathing tests as usual, as I've described before a 3-hour exposure to isocapnic hypoxia, not just 20 minutes once a day, a 3-hour. And then we measured again. And then we thought, do the effects linger? Well, let's test again 1 hour afterwards. We did this with the help of David Cunningham in his laboratory at the University of Western Ontario. And Safraz Mohammed did this one as well as the one previously. How did we do it? They have a hypoxic chamber. Here's Brad Hansen standing in the doorway. And this hypoxic chamber, well, we can flow in nitrogen or oxygen in order to keep the PO2 where we want it. Well, how do we control the CO2? Because CO2 is going to accumulate. We have a happy subject sitting there for 3 hours producing CO2. And we can monitor with nasal prongs what his PO2 end tidal and PCO2 end tidal is, so we can keep him isocapnic and hypoxic or normoxic, whichever we want. But how do we control the CO2? Well, you see these large tubes over here and this great big box here. This is a CO2 absorbing unit, and we have a fan, and we control the speed of the fan, so we can control how much flow goes through here, therefore how much CO2 we remove. And this is a LabVIEW program. We'll talk about this next week. That controls the chamber itself. What did we find out? To make a long story short, here is the hyperoxic test, PO2 of 150, and that's the central chemoreflexes. And here is the start in black, 3 hours later in blue, 1 hour after that in red. They're virtually the same response. But if we look at the hypoxic test now, hypoxic rebreathing and going to involve the carotid body response. Here we are at the start. 3 hours later, the ventilatory recruitment threshold has moved down. And 1 hour later, it's still down. So ventilatory recruitment decrease. That means we have a tonic increase in carotid body activity over that 3 hour period developing. And we looked at the group responses here to say what happened to the ventilatory recruitment threshold as a percent of baseline to normalize everybody. And yes, it went down after the 3 hours and stayed down. Well, are you sure that's not just because they were sitting in a confined space for that long and that did something to them? Well, we had to sit a few of them in there for 3 hours in normoxia. And we saw that, in fact, if anything, these are the individual responses in the thin lines, but the overall response, if anything, the ventilatory recruitment threshold went up, not down. So here's the summary. Again, this is a nice picture from Peter Robbins' lab to show what happens with ventilation if you have isocapnia and if you don't have isocapnia, which is called poikilocapnia. PCO2 is free to change as you apply the hypoxia. So here's the time over hours in their case that they applied. Here's the initial, and we're going to look at this one that I've outlined in red, which is isocapnia is maintained. We see hypoxic ventilatory decline, but after that, as we saw in the 3 hours, we begin to increase ventilation. And as the hours proceed, it gains more and more. So that increase in carotid body activity, as we measured by the ventilatory recruitment threshold, continues to change. However, if we allow PCO2 to change, what kind of response do we get? Well, we do get a response, but the CO2 decreases. That simply withdraws our CO2 stimulus. So even though we may have begun to release the carotid body from its normal oxygen level, which suppresses it, so this is fail-safe, nevertheless, because we have done that and got an increase in ventilation, we've decreased the PCO2 and we've drawn it. So if you look at that, we don't really get much of an increase in ventilation at all. So for altitude, in order to get the ventilation we need, we have to look longer than this for further development in what is happening to the carotid body. So, lesson 3, changes in the hypoxic ventilatory response are the result of changes in the ventilatory recruitment threshold of the carotid body chemoreceptors. We didn't see changes in our hypoxic rebreathing tests, only in the hypoxic. So it's carotid body. The ventilatory recruitment threshold increases for hypoxic ventilatory decline and decreases for the tonic activity potentiation that we see with intermittent hypoxia and with prolonged hypoxia. So, do they reflect a change in the response threshold of the actual sensor, which is sensing hydrogen ion concentration? Are there acid-base compensation changes involved in this? Some questions to consider, which we don't really have good answers for. Here's a sort of theory that I have in my mind that may help understand what is happening. We know how the carotid body works. The glomus cells manufacture transmitter substances which are stored and then released, and then they activate the carotid body sinus nerve endings. So transmitter vesicles release from the stored vesicles when we get hypoxia. We say, oh, yep, dump a few out and stimulate. We have a brisk initial response. But those stores become depleted over the first 20 minutes. So that is the response declining, hypoxic ventilatory decline. But we slowly begin to upgrade the manufacture of transmitter production if we maintain that hypoxia, or we stimulate it by repeated exposures. And so we see the hypoxic ventilatory response increase. And that's we've observed in the ventilatory recruitment threshold change. And even over a long time, we see carotid body hypertrophy because of HIF1 alpha. Again, that is a fail-safe system, as I've talked about. It's usually kept under control, but if we get hypoxic, we don't destroy it, and we start to accumulate it, and it does its job. Finally, altitude hypoxia itself, again, an example of potentiation. We have three important additional changes to consider with really long exposures to hypoxia. We have a shift left in the oxygen dissociation curve. That's because there is a decreased arterial hydrogen ion concentration due to the elimination of PCO2 with our increased ventilation. We have that hypocapnia and decreased arterial hydrogen ion concentration. We get a renal compensation activated. We get brain acid-base control compensation activated. We get a cerebral blood flow change. And they may affect the central chemoreceptors, or maybe not. That's up for debate. Does cerebral blood flow? It'll increase to the rest of the brain, but does it increase to the area, the retrotrapezoid nucleus area where the central chemoreceptors are, or not? And we may develop ventilation-controlled instability, especially in adapting sea level residents, because we've tuned up the response to CO2 because we're now hypoxia in the carotid body. Here's somebody on Mount Everest making some experimental measurements here. Amazing. So Ron Samoji took charge of this investigation and looked at subjects over 5 days at altitude and afterwards. Again, we did modified rebreathing tests, and we did it before they went to altitude at sea level, SL, at altitude after 5 days up here at 3480 meters. And then 6 weeks later, we said, oh, any hangovers from this? And we measured them again. So it provided us with some interesting data. Here's the kind of experimental apparatus and setup that you have in a cramped hut at altitude, where you can actually make experiments and do rebreathing in out of the bag, as Ron is doing here, or looking at this information here on the computer, running, I think, yes, one of my programs here. So, altitude exposure for 5 days, what happens? Sea level, altitude. This is the hyperoxic test. The ventilatory recruitment threshold has gone down. You see, the sensitivity hasn't changed. It's the ventilatory recruitment threshold that's gone down. So we get, for any given CO2, we get a higher ventilation. We see in the hypoxic rebreathing tests also this shift in the ventilatory recruitment threshold down. So why has this occurred? Well, we think this is definitely due to something that's happening in the carotid body, the increase in the tonic activity, which is providing us with our ventilatory drive to cope with the fact that we need to breathe more at altitude. But this actually may be due to the fact that we're measuring this according to end-tidal PCO2, and is the brain responding to the brain hydrogen ion concentration? Has that changed because the brain is now at a lower PCO2 and therefore at a lower hydrogen ion concentration? That may have shifted. I'll show you how that might occur for the cerebral blood flow response to CO2. So if we corrected this, would we correct? But I don't think we would correct this. And indeed, it's the ventilatory recruitment threshold that changes that we see in the hyperoxic test. This is the group response. Yes, definitely decrease. This is the threshold itself in millimeters of mercury PCO2, down significantly. Back up again, mostly 6 weeks later. Was it 6 weeks later? Yes. But the hypoxic rebreathing ventilatory recruitment threshold went way, way down for a much further decrease. And by the time 6 weeks had gone by, it still wasn't back to normal. We had induced that extra activity, tonic activity in the carotid body, and it's still there later. Not significantly lower, but I think it would be if we had more, if we'd done it a little bit earlier, perhaps than 6 weeks, or more subjects. Now, what about individual populations? What has happened to them? They've been born and they live at altitude, and Marat Slassarev took on these expeditions and the experiments done there, in this case in Andean highlanders and lowlanders that went there for 10 days acclimatizing. You can see the lowlanders are much more sensitive, whether it's a hypoxic test or a hyperoxic test. And what has happened to the Andeans? Oh, they are quite relaxed. Their sensitivity to CO2 is much less. Their ventilatory recruitment thresholds are about the same, however. Why is that? Probably because they, like us, need to control their hydrogen ion concentration at reasonable levels for enzymatic and cellular activities. However, here we have people up in the Himalayas. They're a bit different than the Andeans here. They are reasonably sensitive. I've got it the wrong way around. They are about the same, the highlanders, as the Andeans. So this seems to be something that in prolonged hypoxia, because you live there, you become desensitized to the carotid body and the central chemoreceptor response to CO2. So you don't bother increasing your ventilation so much. But they live now at a lower PCO2. But again, if we corrected that to their normal hydrogen ion concentration, we'd find probably they are normal. And so here's our lowlanders here, quite sensitive, and not the highlanders here. Why has that occurred? That is the change in the genetic makeup of this population. And what we would have liked to have done is studied the Ethiopians who have lived at altitude for much longer periods than either the Himalayan or the Andean highlanders, but we never made it there. So there are quite a few papers around on the genetic changes that occur with altitude adaptation. So that's another aspect to remember and be concerned about. What about the oxygen dissociation curve? We mentioned earlier that we have a left shift in that. Well, we are the first lab to actually look at an in vivo oxygen dissociation curve of people that have gone to altitude, and that was done by Dalia Balaban as her project. You'll notice that Marat is there, and a few other people that from the Joe Fisher lab. And we measured the in vivo oxygen dissociation curve, and I fitted a model to it. And we found that it is left shifted. Here's what we published, the P50 versus the pH. The P50 is the partial pressure of oxygen at which you find a 50% saturation. So it is looking at these here for the various populations that we studied, various groups. And you can see there's a lovely linear relationship between the pH, the hydrogen ion concentration, and the P50. So that lowlanders are here, highlanders, yes, here. And if we did a PO2 of PCO2 of 35, a PCO2 of 40, Isocapnic, and PCO2 of 45, and we measured the oxygen dissociation curve, what would we see? So the lower your PCO2, the further you shift. And that is what's happening in lowlanders versus highlanders. What does that lift shift do for you? It's the same in animals, possibly the Himalayan and Ethiopian highlanders, and it favors loading oxygen onto the hemoglobin at the lung. And that's the same as animals do. But it solves the debate, because people have been saying, no, it should go right shift so that we unload oxygen easier at the tissues. No, it should go left shift so that we take up oxygen more easily. Well, it's a left shift. The other argument that went on that was this adaptation, this shift was due to the 2,3-DPG molecule, but we found that no, it is simply due to the change in pH. What about altitude exposure and cerebral blood flow? And this comes from Mickey Phan's experiments that were done in Rob Roach's lab and published here. One of the things they did is they took arterial samples at sea level after 1 day and then 16 days at altitude. And they also measured the hydrogen ion concentration, and they plotted versus PCO2, so they had the CO2 dissociation curve relationship. And note, this is the arterial hydrogen ion concentration. The central chemoreceptors behind the blood-brain barrier, we don't know what happens very well with that. Central acid-base regulation occurs, we're sure, but how much? And this is what I was talking about before, about whether the central key receptors, for example, whether their ventilatory recruitment threshold shift is due to the low CO2 and the difference between CO2 and the actual stimulus of hydrogen ion concentration. Well, here we measure the response to CO2 of cerebral blood flow measured by the middle cerebral artery velocity with a Doppler probe. All of the responses for each individual are drawn in the dotted lines, and then the average, it's a sigmoidal response. The average is drawn in there. Where their CO2 lay, in each case, is the squares here, and where the midpoint of the sigmoids is the black dots here. So that as we went from sea level and initially went to altitude, we got rid of CO2, we're at a lower PCO2, even lower after 16 days, and we see a shift in the cerebral blood flow response. So the blood flow response to a change in CO2 that we might see becomes greater and greater because we've shifted it down as we stay at altitude. But why don't we use what we saw in this relationship between PCO2 that we measured, and we, instead of plotting the middle cerebral artery velocity against end tidal PCO2, roughly arterial, I suppose, against hydrogen ion concentration as determined. And we find that, yes, at altitude, they have gone from normal 40 nanomoles per liter down to 30 nanomoles per liter. So there is an alkalosis. This is maintained over the 16 days. But what has happened to the cerebral blood flow sigmoid midpoints? They now all line up. So that tells you that that's what they are looking at. Altitude, acclimatization, and sleep. Sleep becomes a problem for people going to altitude. It induces what's known as Cheyne-Stokes breathing. And here's an example of what it is here. It's unstable breathing. The wakefulness drive is gone. So if you cross over the ventilatory recruitment threshold, which is now an apnea threshold, if your CO2 goes below that, you stop breathing. So we see that it's gone below the threshold. There's no drive to breathe. They don't breathe. Oh, and then CO2 rises and stimulates them. It's very similar to what you see in obstructive sleep apnea. But this is not obstruction. This is the fact that we've just lost the drive to breathe when we've gone below the ventilatory recruitment threshold or the apnea threshold when you're asleep. And so we breathe, and then CO2 goes down again below the threshold, and we keep on repeating this pattern. And so that is the acclimatization that happens when people first go to altitude. Their sleep is terrible. Is this the hypoxia alone that's doing it? And here's a experiment done in Dempsey's lab. What induces this periodic breathing during sleep in humans? Well, they have given them hypoxia while they're asleep, and they notice the ventilation goes into this pattern here. And so that tells you all about why the system becomes unstable, because the loop gain is increased, because we're hypoxic. We've now turned on the carotid body sensitivity to CO2, fail-safe system. We've removed the oxygen level that is shutting down the carotid body. So now they're fully active, and you bounce back and forth over that ventilatory recruitment threshold because your loop gain is now so high, and you get this instability. The last, I think we're getting near the end, but we have to consider what happens to sympathetic activity in altitude hypoxia. Here's a nice study that shows you what is happening. At sea level, we see the activity. Acute hypoxia, yeah, a bit of an increase maybe. After 10 days at 4,100 meters, and after 50 days, it's even greater. So the sympathetic activity increases. That's got to be from the carotid body increase in their tonic activity, in my opinion. And finally, chronic hypoxia, as I've said before, it's fail-safe. If you have oxygen, HIF is destroyed. It's made continuously and then destroyed. If you lose that destruction because the oxygen is too low, then we get HIF1 alpha effects. So you can adapt to moderate and short exposures to hypoxia, but we lose that with increasing severity and duration. We noticed that in the Highlanders, where they had lost a lot of their response to hypoxia and even CO2. And so what do? They have this mechanism, the molecular genetic mechanism of HIF1 alpha pathway, and it orchestrates the genes for cells, tissues, organs, and body responses to hypoxia. Lots of regulatory pathways triggered and so forth, and you can read all about that. And of course, Greg Semenza received the Nobel Prize for that in 2019. And I note, finally, that all of these compensatory mechanisms aims to optimize oxygen delivery and therefore protect the brain and the heart from hypoxic injury. If we look at cerebral blood flow and see if we have chronic hypoxia, such as in anemic patients, in sickle cell disease patients, what happens to them? Their cerebral blood flow increases as the oxygen content goes down in this curved relationship. That's from John Wood's laboratory. And this is where I made the fail-safe model that I've introduced you to before. And if you look at the way that that model works, in that it is a smooth muscle cell trying to regulate its internal hydrogen ion concentration, and when that fails because the oxygen supply has failed, it loses its ability to maintain tone and cerebral blood flow increases. And that model works very well to predict what is going on. Whether you reduce the partial pressure of oxygen, such as going to altitude, whether you have a problem because you have hemoglobin loss, anemia, or hemoglobin that doesn't work very well, sickle cell disease. Either way, you get this relationship, and this is the relationship, and it fits very well with this.